I've been asked to talk about three different topics: effective exercise on insulin action, effective calorie restriction on insulin action, and effective aging on insulin action. Now, any one of those topics would be difficult to cover in 1 hour. Want to hold both of them? So I'm only going to be able to hit a few high points. I'm going to start out by talking a little bit about aging. I think it's important to think about aging in terms of primary and secondary aging. Primary aging is the inevitable deterioration in structure and function of cells and tissue with age that occur independent of disease. Whereas secondary aging is the deterioration in structure and function of cells and tissues caused by diseases, trauma, lifestyle factors. And so secondary aging is, at least to some extent, preventable. Two terms that tend to be confused are life expectancy and lifespan. Life expectancy is how long an individual can expect to live at birth. And there's been an enormous increase in life expectancy over the last 200 years as a result of improvement in public health measures such as cleaning up the water supply, cleaning up the food supply, vaccination against childhood diseases, antibiotics, and so on. So obviously, life expectancy can be markedly modified by environmental factors. Whereas lifespan is primarily determined by genetic factors. It's how long, the maximal period of time that a species lives. And the genetic component is obvious from comparisons of rats and mice with humans. A rat has a maximal lifespan of about 4 years, whereas with humans it's about 110 years. So whereas life expectancy can be markedly modified by environmental factors, lifespan is largely determined by genetic factors. However, it is possible to somewhat modify maximal lifespan. The intervention that's been studied most extensively is calorie restriction. And it's been known for over 80 years now that if you reduce a laboratory rat or mouse's food intake by about 25 to 40%, they end up living 25 or 30% longer. A variety of other interventions that result in downregulation of the insulin, IGF-1, mTOR signaling pathway also has been shown to increase maximal longevity in laboratory rodents. This increase is relatively modest. It's about 20%. Let me give you an example of what I've been talking about. About 25, 30 years ago, I did a series of studies in which I looked at the effect of exercise on longevity. This study was done on male rats, which are very unusual in that they don't increase their food intake to compensate for the increase in energy expenditure. All other species that I'm aware of increase their food intake to compensate for the increase in energy expenditure with exercise. Even female rats do. But for some reason, male rats don't. So they end up, if you start exercising a mouse or a rat when they're young, they end up being much smaller than a freely eating sedentary animal. This line here is the sedentary food-eating animals, and this is the exercise group. And you can see that the exercise groups average longevity increased considerably, whereas there was no increase in maximal longevity. In contrast, the animals that were food restricted to keep their body weight the same as that of the exercisers showed the usual increase in maximal longevity. And the food restriction required to keep these animals at the same weight as the runners was about 30%. And you can see how much smaller the food restricted and exercised animals were than the sedentary controls, which were just sitting in their cages and with unrestricted food intake. Now, this effect of exercise has been referred to beneficial exercise on longevity. I think that's the wrong way of looking at it. Exercise is normal. I mean, throughout evolution, it was necessary to exercise in order just to stay alive. And I think that the exercising animals are showing normal aging, normal longevity. And what the exercise is doing is protecting against the harmful effects of a sedentary lifestyle with unrestricted access to food. So the sedentary rats are representative of what's happening to a large proportion of the population in the United States that are not exercising and are getting very fat. Now, the finding that the exercising male rats did not live as long as the food-restricted male rats came as a surprise to a lot of people because the relative energy deficit was the same. And actually, the exercised animals were leaner than the food-restricted animals. So it was suggested that maybe the exercise was having a harmful effect, maybe increased free radical damage, or increased oxidative stress that was counteracting the beneficial effect of reduced availability of energy for growth. So we did another study in which we had a group of exercised animals that were food restricted. And as you can see, the food restricted exercising animals lived just as long as the animals that were kept sedentary and food restricted. So exercise does not have a harmful effect that prevents the beneficial effects of an energy deficit. I think this shows that the effect of calorie restriction is mediated by a decrease in food intake, not by the relative negative energy balance. Now, as I mentioned, mammalian evolution occurred in the context of vigorous physical activity, both to get food and to avoid becoming food for other predators. And the selective pressures dictated by hunter-gatherer lifestyle has shaped the human genome over the last 2 million years. So we're therefore poorly designed. In other words, we're genetically maladapted for the sedentary lifestyle that's the result of the enormous societal changes that have occurred as a result of industrialization and technological advances. This is what normal humans look like. These are a group of Amazonian Indians. This picture was taken about 50 years ago when there were still some hunter-gatherer groups. You can see that these individuals are well-muscled and extremely lean. I would guess that the average body fat is about 8%. And these guys have just caught their lunch. In contrast, this is what a typical middle-aged man in St. Louis, where I live, looks like. We're having a huge epidemic of overweight and obesity. And if any of you have been in an airport in Chicago, St. Louis, Atlanta, or any of the other southern states, you'll see dozens of men walking through the airport looking like this, or even more obese. This type of obesity, the central obesity, as you know, is associated with the so-called metabolic syndrome, the central feature of which is insulin resistance, which, if it persists long enough, usually progresses to type 2 diabetes. Now, it's generally thought that glucose tolerance deteriorates with age. And I'm sure that it does to some extent, because everything deteriorates with age. But I think that the effect of aging on insulin action has been grossly exaggerated. What I'm showing you here are glucose tolerance curves on about 87 men and women in the 65 to 75 year age range, compared to those of healthy young individuals. These are the young individuals, and these are the older individuals who have perfectly normal glucose tolerance. These older individuals have high normal. These are impaired glucose tolerance and full-blown diabetes. This is a separate group which is lumped with type 2 diabetes, but these individuals really have insulin deficiency and are not insulin resistant. Now, this is the glucose tolerance of the healthy young individuals, the insulin levels, and these are the insulin levels on the older people who have perfectly normal glucose tolerance. And you can see that the insulin levels are higher, indicating that they're somewhat insulin resistant. But these older individuals who have normal glucose tolerance and slightly elevated insulin levels are much fatter than the young people. They have about 23% body fat, whereas young individuals had about 16% body fat. And as I'll show you later on in the talk, older individuals who have managed to stay lean, either by means of exercise or by means of calorie restriction, have glucose tolerance that looks just as good as that of the young individuals here. The commonly used model of abdominal obesity resulting in insulin resistance is a laboratory rodent fed a high-fat diet, either rats or mice. And the development of insulin resistance and abdominal obesity is much more rapid. I mean, everything is compressed because of their shorter lifespan. And you can see that just within 8 weeks, there's a large increase in intra-abdominal fat. And concomitant with that, there's an increase in insulin resistance. What I'm showing you here is the results of a euglycemic clamp, in which blood glucose was clamped at 110 milligrams and insulin levels remained at 100 microunits per milliliter. And you can see that within 4 weeks, these animals were already insulin resistant, and at 8 weeks, they were severely insulin resistant. And as I showed you in the previous slide, they had already gained a lot of intra-abdominal fat. Now, doing clamps or hindquarter perfusions in rats is very expensive. So what we usually do is just measure glucose tolerance in an isolated muscle preparation. And the muscle that we usually use is called the epitrochleus. It's a very thin muscle of the forelimb that's thin enough so that oxygen diffusion and sugar diffusion is not a problem. We also sometimes use the soleus, but the soleus is too thick for adequate oxygenation. So we split the soleus and use the split soleus muscle. What I'm showing you here is the rate of a non-metabolizable glucose analog, 2-deoxyglucose, take up by one of these muscle preparations, and an effect of a moderate concentration of insulin and a maximally effective concentration of insulin. At both insulin concentrations, you can see that 4 weeks on the high-fat diet has made these muscles insulin resistant. And if you keep the rats on a high-fat diet long enough, most of them develop full-blown diabetes and get very fat. I'd like to make the point that the insulin resistance is associated with obesity. It's not in fact the high-fat diet per se. It's the effect of increased caloric intake. These are the animals eating the high-fat diet ad lib, and these are animals that are eating the same diet, but in which the diet is restricted to keep them from becoming fat. And you see that the high-fat diet per se has no effect as long as it doesn't result in obesity. Now, in the last few years, people have been questioning the role of intra-abdominal fat. Some people think that intrahepatic fat is more important in causing the insulin resistance, where other people think that accumulation of fat in the skeletal muscles is responsible for the insulin resistance. I don't know what's correct. I mean, there's still a lot of question about whether the insulin resistance is due to products that are being produced by the large fat cells or by the accumulation of fat in the liver or muscle. But if abdominal fat is not responsible for the insulin resistance, it's so closely correlated with insulin resistance that it serves as an excellent marker or endpoint. I think it's very useful. I mean, it's quite difficult to measure liver fat or muscle fat, whereas it's very, very easy to measure somebody's abdominal circumference, or just look at them. What I'm showing you here are some magnetic resonance images taken across the abdomen. These are not very good ones. This was a study that was done quite a long time ago. In this study, we exercised a group of middle-aged individuals with mild diabetes vigorously for about 6 months. They're supposed to exercise 5 days per week, and they averaged about 4 days a week. And they ended up losing 5-6 kilos of fat. And I think you can probably see that a lot of this fat has disappeared. And look what's happened to their glucose tolerance. It's gone from mild diabetic to normal, despite a decrease in insulin levels, showing a huge improvement in insulin action. Something that concerns me a lot now is that when an individual is diagnosed with type 2 diabetes, he's immediately put on anti-diabetic drugs and treated as though he's got a lifelong disease. And for most people, it is a lifelong disease. Whereas in the early stages, this is a completely reversible process, a curable process, either by calorie restriction or exercise. This is from a more recent study in which we compared the effects of severe calorie restriction, a 25% reduction in calories, or exercise that resulted in a 25% increase in calorie expenditure. And this shows the results on a lady who was very compliant with the diet, and she lost about 16 kilos. And you can see that all this fat is gone. Then a huge decrease in mental and other intra-abdominal fat. Now, this lady had mild diabetes, and you can see that at the end of the 12-month period, she's got excellent glucose tolerance and low insulin levels. Her diabetes is cured. Now, it's usually thought that you can prevent obesity by means of exercise, but you can't reverse it. And the reason for that is that most middle-aged people, particularly obese people, have a very low exercise capacity, so that in order to burn a significant number of calories, enough calories per day to lose a significant amount of weight, involves a huge amount of time. Nevertheless, it is possible. And what I'm showing you here are the abdominal magnetic resonance images on a man who was very compliant with the exercise program. And you can see that he's lost a huge amount of both subcutaneous and intra-abdominal fat. And this individual also lost about 16 kilos. But in order to do so, initially he was exercising 2 hours a day, walking. But as his fitness improved, he was able to exercise harder, so that by the end of the year he was only exercising about 1 hour a day to burn the same amount of calories. So exercise results in weight maintenance, weight loss, and prevents and can reverse insulin resistance by means of preventing or reversing abdominal obesity. I keep saying abdominal obesity may also be reversing fat accumulation in the liver and muscles. Now, in addition to the effect of weight loss, exercise has another effect that results in an improvement in insulin action in a very short period of time. In this study, we had a group of obese diabetic men walk on a treadmill for an hour a day for just 7 days. And you can see that their glucose tolerance has gone from diabetic to impaired in just 7 days. And a large decrease in insulin levels, too. Now, 7 days of exercise is much too short a period to result in significant weight loss. So obviously something else is going on. And actually there are two things going on. An acute bout of exercise is followed by a large increase in insulin sensitivity. And there's also a big increase in insulin responsiveness. And I'll show you what I'm talking about in a minute. But these are very short-term effects of exercise. They wear off very quickly. As I've shown you in the previous slides, exercise results in a reduction in insulin levels. And I think that this adaptation was selected to prevent the development of hypoglycemia during exercise. But it's a very consistent effect. What I'm showing you here is the effect of a single bout of exercise on insulin sensitivity. In this study, rats were exercised by means of swimming, and here we measured glucose uptake in response to a very low concentration of insulin in the epitrochleus muscle 3 hours after the exercise, when the acute effect of exercise on glucose transport had worn off. And you can see that there's a huge increase in insulin sensitivity. Insulin sensitivity is defined in terms of the concentration of insulin that results in a half maximal effect. There's no improvement in response to a maximally effective concentration of insulin. Increase in sensitivity only is evident at low concentrations of insulin. Now, this is a very, very transient effect, which lasts just long enough to permit glycogen repletion to the pre-exercise level. And as the effect of exercise on insulin sensitivity is wearing off, it's replaced by an increase in glucose transporter protein, GLUT4 protein. The glucose transporters are located within the muscle cells, and in response to insulin or to exercise, they move to the cell surface, and they're responsible for transporting glucose into the cell. And the increase in transport is proportional to the increase in GLUT4. And you can see that with exercise, there's a very rapid increase in the GLUT4 protein, which results in a proportional increase in insulin responsiveness. Insulin responsiveness is defined in terms of response to a maximally effective insulin concentration. And an increase in insulin responsiveness results in a shift in the glucose-insulin relationship upwards. At all insulin concentrations, you have a greater increase in glucose uptake. This effect, the increase in GLUT4 and in glucose transporters and insulin responsiveness, is primarily responsible for the rapid improvement in glucose tolerance in response to exercise. But in order to maintain it, you've got to exercise at least every other day or every third day, because it wears off very quickly. The increase in GLUT4 is gone within about 3 or 4 days, unless you do severe carbohydrate restriction and prevent glycogen depletion. So there's a very transient effect. I've been showing you data on rats, but exactly the same thing happens in humans. What I'm showing you here is the increase in GLUT4 protein in the quadriceps muscle of a group of young men and women who trained hard on cycloergometers for about 7 days. We just took muscle biopsies of the quadriceps, and you can see there's a huge increase in GLUT4 within a week. I'll move on now and talk a little bit about calorie restriction. About 9 years ago, I got an email from a man who said that he was a member of the Calorie Restriction Society, and he wanted my advice on what kind of exercise he should be doing in order to try to maintain some muscle mass. Now, I didn't know there was such a thing as a Calorie Restriction Society, and my colleague Luigi Fontana was working with me as a postdoctoral fellow at that time. So I said, Luigi, why don't we contact these people and see if we can't study some of them? So through this individual who had sent me the email, we contacted the Calorie Restriction Society, and over the years, we brought about 45 of them to St. Louis, some of them repeatedly, and obtained a considerable amount of information on them. It turns out that these individuals are sort of disciples of Roy Walford, who published a book called The 130-Year Diet, in which he postulated that the effects that he saw in his calorie-restricted mice also were relevant to humans, and if humans were calorie-restricted, they could live to be 130. And these people believed him, and they're doing severe calorie restriction with optimal nutrition. What I'm showing you here is some data that we obtained on the first 18 of these individuals. Something that's very strange and that I have no explanation for is that nearly all the members of the Calorie Restriction Society are men. So far, we've only been able to find 6 severely calorie restricted women. 4 were in this first group here. And you can see that these individuals are very thin and have very, very low body fat. Even middle-aged, competitive middle-distance runners, master athletes, are not this thin. And the thing that I found most amazing was the extremely low blood pressure. The average blood pressure is 103/63. That's the sort of blood pressure that you see in little kids. They have low cholesterol, low LDL cholesterol, remarkably high HDL cholesterol, a low total to HDL cholesterol ratio, and very low triglyceride levels. They also have low fasting glucose levels, very low fasting insulin levels, and they also have very low levels of inflammation. TNF-alpha, and the C-reactive protein is barely measurable. They also have low level of transforming growth factor beta and low thyroid hormone levels. This T3 level is right at the bottom level of normal range. And recently we found that they also have a low body temperature. We measured central body temperature. Now, a problem with this sort of cross-sectional study is that people always raise the question, what were these people like before they started on the calorie restriction? Are these a very special or unusual group of people? Unfortunately, some of them went to their personal physicians before they started on the calorie restriction and had a medical examination, and then went back after a year to see what had happened. And we've been able to get the physicians' records on these people, on about 14 of these individuals. And you can see that after about a year, in the first place, these are very average individuals. The cholesterol level, LDL cholesterol, HDL cholesterol, triglycerides, blood pressure, all right in the mid-range for middle-aged men. And you can see that after 1 year of calorie restriction, there's been a huge weight loss, big decrease in cholesterol, big decrease in LDL cholesterol, a large increase in HDL cholesterol, big increase in triglycerides, and a remarkable decrease in blood pressure. And these are the values that we got on them when we studied them, which is about 6.5 years later. And you can see that there's been a further weight loss, further decrease in LDL cholesterol, and a further increase in HDL cholesterol. This is a huge increase in HDL cholesterol from 47 to 64. I don't know of any drug regimen that you could put an individual on that would do this. There's also a really huge decrease in triglyceride levels from 134 to 49, and this big decrease in blood pressure that I mentioned earlier. So these are not special individuals. I mean, what we are seeing are the effects of calorie restriction. We've also compared the calorie-restricted group with a group of competitive middle and long-distance runners. These are master athletes who are involved in age group competition. And then we have a so-called Western diet group that are eating usual diets that are healthy and not overweight. And you can see that the exercisers are just about as lean as the calorie-restricted group, but are considerably heavier as a result of a much larger lean body mass. Calorie restriction results in loss of both fat and muscle. The calorie-restricted group, as you might expect, have elevated adiponectin levels. As I mentioned before, they have very low levels of inflammation. Their IL-6 levels and TNF-alpha levels are much lower than that of the Western diet group. The exercise group also has low IL-6 levels, but the calorie-restricted group has much lower levels of C-reactive protein than the exercise group, although the exercise group is much lower than the sedentary controls. So in terms of inflammation, regularly performed exercise also has a beneficial effect. We've obtained glucose tolerance on some of these individuals. I think there were 28 individuals in this group. And we were very surprised to find that the calorie-restricted individuals did not have very good glucose tolerance. You can see that the 120-minute value is even higher than that of the sedentary people on the Western diet, whereas the exercising group has glucose tolerance is just as good as that of healthy young people. And this was very unexpected because the calorie-restricted individuals have very low fasting glucose and insulin levels. And you can see that their insulin levels are also higher than those of the exercisers, but obviously not as high as those of the group on the Western diet. I'm looking at the data on these individuals. It was clear that they fell into two groups. 17 of them had excellent glucose tolerance, whereas 11 of them had impaired glucose tolerance. These are the 18 who had normal glucose tolerance, and at average age 53, their glucose tolerance is as good or better than that of young individuals, whereas the other levels had clearly impaired glucose tolerance. And the question, what's responsible for this huge difference? And at this point, we're not sure. They're the same age, and based on their diet records, the caloric intake is the same. But clearly there's a big difference between their diets because the ones with impaired glucose tolerance have a much higher fiber intake, suggesting that all of the calories that are estimated calories are not being absorbed probably. They're also in less good physical condition. Their oxygen uptake capacity is much lower, and they're much thinner. The group with a good glucose tolerance on average had a BMI of 20, which is at the lower range of normal, whereas the ones who had impaired glucose tolerance were quite emaciated. I mean, these individuals really look like concentration camp victims. They're very low lean body mass, low IGF-1 levels, very low testosterone levels, and very low triiosocyanine levels. So my interpretation is that the calorie-restricted individuals with impaired glucose tolerance are really overdoing the calorie restriction. I think it's not just calorie restriction, I think it's semi-starvation. We've obtained muscle biopsies on 15 of the members of the Calorie Restriction Society. And there's some downregulation of the insulin IGF-1 signaling pathway. As you can see, AKT, PKB is downregulated about twofold. Now, we don't have glucose tolerance data on these 15 individuals. These two studies were done about 5 years apart. And what I suspect is that this is a composite of all 15 individuals. I suspect that the ones who are extremely severely calorie restricted, the ones that have impaired glucose tolerance, probably have a much greater downregulation of the insulin signaling pathway than the ones who don't. And this is something that we're going to have to check out in future studies. So in conclusion, and I think this is probably the most important message, is that abdominal obesity, metabolic syndrome, is completely reversible by means of negative energy balance induced either by diet or exercise. And type 2 diabetes is also reversible by means of negative energy balance in the early phase before there's severe damage to the pancreatic beta cells, which occurs as diabetes progresses. Long-term severe calorie restriction results in excellent insulin action and glucose tolerance, whereas extremely severe long-term calorie restriction results in impaired glucose tolerance. As I mentioned, Luigi Fontana started this study and has been primarily responsible for it. Other members of our group who have been involved are Dennis Villarreal, Tim Meyer, and Sam Klein. And my other colleague, Dr. JohnO Han, has been responsible for the biochemical and molecular biological measurements. And that's my last slide. Thank you.